Good morning, good afternoon, good evening, depending on where you are physically located. Thank you for joining Synchrogenics and RAPS for this webinar. My name is Nellie Forwood, Principal Regulatory Writer at Synchrogenics. The objectives of this webinar today are to learn about the different types of impurities that are in pharmaceutical products. We're going to understand the process for handling these impurities in drug substances and in drug products, according to the regulations and guidance documents. We're going to review the decision trees for the impurities, and we're going to present some case studies and based on our experience with some impurities. So what is an impurity? An impurity is any component of the drug substance or drug product that is not the chemical entity, is not defined as the drug substance or an excipient. Basic impurity categories are organic or process and drug-related impurities, inorganic impurities, elemental impurities, residual solvents, and nitrosamines. Currently, the establishment of drug substance and drug product impurity acceptance criteria can be supported by clinical data, non-clinical data, comparative impurity analysis of the proposed drug product with an FDA-approved drug product, analytical precision of the method used to measure the impurity, and manufacturing process capabilities. There are a couple pages of definitions here, but we're going to go over three of them, which are the identification threshold, which is the limit above which an impurity should be identified, the qualification threshold, which is the limit above which an impurity should be qualified, and by qualified we mean the process of acquiring and evaluating data that establishes the biological safety of an individual impurity or a given impurity profile at the level specified. And we also have the reporting threshold, which is the limit above which an impurity should be reported. If you have an impurity that is below the reporting threshold, according to the guidance documents, you do not need to report it, nor is it added to your total to make up your total impurities. So organic impurities can arise during the manufacturing process and or storage of the drug substance and drug product, and can be identified or unidentified, volatile or non-volatile. And these include starting materials, byproducts, intermediates, degradation products, reagents, ligands, and catalysts. Inorganic impurities can result from the manufacturing process and are normally known and identified. They include your reagents, ligands, catalysts, heavy metals, and other residues, inorganic salts, and other materials such as filter aids and charcoal. They are normally detected and quantified using pharmacopoeia or other appropriate procedures, and they can be a carryover of catalysts to the drug substance and or drug product and should be evaluated during development. Residual solvents are organic volatile chemicals that are used or produced in the manufacture of the drug substance, the excipients, or in the preparation of the drug product. They require the control of the residues and solvents are used in the manufacturing process. This should be discussed and presented according to ICH Q3C and also USP 467. Residual solvents are split into three classes. Class 1 solvents, which are solvents that you need to avoid at all costs. Class 2 solvents, which are solvents that may be used to a limited extent if you absolutely have to. And Class 3 solvents, which are solvents with a low toxic potential. All are typically determined using chromatographic techniques such as gas chromatography. Another type of impurity is elemental impurities. These may arise from several sources. They may be residual catalysts that were added intentionally in the synthesis process, or they may be present as impurities. They are also placed in the three classes based on the toxicity and the likelihood of occurrence in the drug product. Class 1 are human toxicants that have limited or no use in the manufacture of pharmaceuticals. Class 2 are generally considered as route-dependent human toxicants. Class 2 is divided up into Class 2A and Class 2B, a relatively high probability of occurrence in the drug product and reduced probability of occurrence in the drug product. And then Class 3 elemental impurities are have relatively low toxicities by the oral route of administration, but they may require consideration in the risk assessment for inhalation and parenteral routes. Nitrosamines are a new, considered new compound that they've been, in June 2018, FDA was informed of the presence of an impurity identified as N-nitrosodiethylamine, NDMA, in the ARB valsartan. Through investigation, the agency determined that numerous lots of valsartan and a few other ARBs products from different manufacturers contained unacceptable levels of nitrosamines. The drug product manufacturers voluntarily recalled the affected batches of these drug products, which led to a drug shortage in some of the affected products. In addition, FDA evaluated processes that use common amines in API synthesis and learned that common synthetic pathways could also introduce other types of nitrosamine impurities besides NDMA. In September 2019, FDA learned that some common heartburn products, rhinantidine, commonly known as Zantac, and nizantidine, commonly known as Axid, contained unacceptable levels of NDMA. FDA recommended that manufacturers voluntarily recall rhinantidine and nizantidine products with NDMA levels above what the agency considered acceptable. Recently, preliminary findings from FDA stability testing raised concerns that NDMA levels in some rhinantidine products stored at room temperature can also increase with time to an unacceptable levels. FDA's preliminary results using accelerated stability testing demonstrated that elevated levels of NDMA were measured in all products after 2 weeks. FDA's testing suggested that NDMA levels increased with storage over time. On April 1st, 2020, FDA requested that all rhinantidine products be withdrawn from the US market. In December 2019, FDA became aware that some metformin diabetes medicines in other countries were reported to have NDMA. In light of this information, FDA acquired samples of metformin to test for NDMA. By February 2020, the agency had identified NDMA in some samples, but did not find levels exceeding the acceptable intake limit. In May 2020, further FDA testing revealed that certain lots of metformin extended release formulation contained NDMA above the agency's recommended acceptable intake limit. Based on that testing, FDA requested that identified applicants voluntarily recall these lots of the extended release metformin. FDA continues to investigate possible NDMA impurities in metformin and other drug products and will advise companies on the appropriate action. Last week, the FDA had a workshop on the Monday and Tuesday, the 29th and 30th of March, where they had several questions that they asked various nitrosamine experts throughout the globe. I sat in on this session and found it very interesting. It was very, very intense. Okay, so what do you do when you find an impurity? Today, we're going to go through these various steps. You determine the highest daily dose of your impurity, and you need that for ICH M7, Q3A, and Q3B. You're going to assess if the impurity is possibly mutagenic. That's according to ICH M7. Then you're going to develop the identification and qualification thresholds for that, according to ICH Q3A and Q3B. You're going to look at residual solvents, possibly in the drug substance or drug product, ICH Q3C. You're going to examine potential elemental impurities, ICH Q3D. And then you're going to conduct a nitrosamine assessment for the drug substance and the drug product. And these are according to FDA and EMA guidances that are available. So the ICH M7 deals with mutagenic impurities, and they talk about a threshold of toxicological concern, or TTC. The methods upon which the TTC is based are generally considered to be very conservative, since they involve a simple linear extrapolation from the dose given at a 50% tumor incidence, TD50, to a 1 in 10 to the 6th incidence using your TD50 data for the most sensitive species and the most sensitive site of tumor induction. A value of 1.5 micrograms per day, corresponding to a theoretical 10 to the -5th excess lifetime risk of cancer, can be justified. Some structural groups were identified to be such high potency that intake even below the TTC would theoretically be associated with the potential for a significant carcinogenic risk. This group of high potency mutagenic carcinogens are referred to as the cohort of concern. And this group comprises aflatoxin-like N-nitrosos and alkyl-isyl compounds. We'll discuss the N-nitrosas, which are the nitrosamines that we talked about a couple minutes ago, but we're going to discuss them more in depth later. We're also going to look for established cancer risk assessments, which are based on, the established cancer risk assessments are based on lifetime exposures. So when you're looking at your mutagenic impurities, you're going to look for actual and potential impurities that are likely to arise during the synthesis and storage of your new drug substance and during the manufacturing and storage of your new drug product. The impurity assessment is a two-stage process. First is looking at actual impurities that have been identified and should be considered for their mutagenic potential. And the second is an assessment of potential impurities likely to be present based on your structure of your drug substance, likely to be present in the drug substance, and it's carried out to determine if further evaluation of their mutagenic potential is required. A standard risk assessment of known carcinogens assumes that cancer risk increases as a function of cumulative dose. The TTC-based acceptable intake of 1.5 micrograms per day is considered to be protective for a lifetime of daily exposure. To address less than lifetime exposures to mutagenic impurities in pharmaceuticals, an approach is employed in which the acceptable cumulative lifetime dose, 1.5 micrograms per day, times 25,550 days, is 38.3 micrograms. This is uniformly distributed over the total number of exposure days for your LTL exposure. So if you have you're taking it for less than a month, 1 month to 12 months, greater than 1 year to 10 years, and then greater than 10 years, as is in the table. All right, these steps are applied to synthetic impurities and degradation products are described in sections 5.1 and 5.2 respectively of the guidance. Actual impurities, including those observed in the drug substance above the ICH Q3A reporting thresholds, are looked at. Some impurities below the identification threshold may also have to be identified. And then for potential impurities in the drug substance can include starting materials, reagents, and intermediates in the route of synthesis from the starting material to the drug substance. All right. Risk-based justification should be provided for the point in the synthesis after which these types of impurities should be evaluated for mutagenic potential. For starting materials that are introduced late in the synthesis of the drug substance and where the synthetic route of the starting material is known, the final steps of the starting material synthesis should be evaluated for potential mutagenic impurities. Identification of actual degradation products is expected when the levels exceed the identification thresholds outlined by ICH Q3A and Q3B. Potential degradation products include those that form above the ICH Q3A and B identification thresholds during accelerated stability studies, which are typically 40 degrees C, 75% RH for 6 months, and confirmatory photostability studies as described in ICH Q1B. Knowledge of relevant degradation pathways can be used to help guide decisions on the selection of potential degradation products to be evaluated for mutagenicity, from degradation chemistry principles, relevant stress testing studies, and development stability studies. Actual and potential degradation products likely to be present in the final drug substance or drug product and where the structure is known should be evaluated for mutagenic potential. And post-approval, submissions involving drug substance chemistry manufacturing controls should include an evaluation of the potential risk impact associated with mutagenic impurities from change to the route of synthesis, reagents, solvents, or process conditions after the starting material. For the drug product, you're going to, which are changes in composition, manufacturing process dosage forms you should include an evaluation of the potential risk associated with any new mutagenic degradation products or higher acceptance criteria for existing mutagenic degradation products. Okay, so this is a table of mutagenic impurities that are continued. There's 5 different classes. Class 1 are known mutagenic carcinogens. Class 2 are known mutagens with unknown carcinogenic potential. Class 3 are mutagens that have an alerting structure unrelated to the structure of the drug substance and with no mutagenicity data. Class 4 are classes that have an alerting structure. It's the same alert as the drug substance or compounds related to the drug substance, which have already been tested and are non-mutagenic. And then class 5 are no structural alerts or alerting structures with sufficient data to demonstrate a lack of mutagenicity or carcinogenicity. And we'll go into this a little bit more in one of the case studies. So going on to ICHQ3A, this is impurities in new drug substances. This guidance applies to drug substances produced by chemical synthesis, not the clinical trial stages or biologics, et cetera. It discusses the handling of impurities should be based on a decision tree, and limits shown in the ICHQ3A attachment 1 for that you need to know the maximum daily dose, and we talked about that earlier. ICHQ3A attachment 3 contains a decision tree for the identification and qualification of impurities. And this is the decision tree. I know you can't read it, but we're going to go into that a little bit more detail in one of the case studies. ICHQ3A also talks about the quantitative results should be presented numerically and not in general terms such as complies, meets limits, et cetera. If you send results to the FDA as complies, they'll come back to you as requesting the actual data. Any impurity at a level greater than the reporting threshold and your total impurities observed should be reported. If your impurity is present at less than 1.0% to 2 decimal, you report it to 2 decimal places. If it's greater than 1.0, you report it to 1 decimal place. Okay, when you're doing any rounding, you should use conventional rounding rules. All right, for ICHQ3B, impurities in new drug products, you want to address only those impurities that are new in the drug product as opposed to the drug substance. Or as classified as degradation products in the drug substance or reaction products of the drug substance, with an exception with an excipient and the immediate container closure system. So anything on the drug substance that may react with your drug substance or drug product container closure system, any reaction products for the drug substance with any of the excipients, and all of that is dealt with ICHQ3B. It also provides reporting, identification, and qualification thresholds for this one also. So you need to know your maximum daily dose. And this is the decision tree for the impurities in new drug products. It's very similar to the decision tree in the ICHQ3A for the drug substance. And again, we're going to talk about this decision tree for the drug substance in one of our case studies. ICHQ3C talks about residual solvents. In addition, for your residual solvents, you're going to need to do fate and purge studies to determine the fate of each of the solvents utilized in your synthesis process. It's only necessary to test for solvents that are used or produced in the manufacture or purification of your drug substance, your excipients, or your drug product. A cumulative method may be used to calculate the residual solvent levels in the drug product from the levels in the ingredients used to produce the drug product. If the calculation results in a level equal to or below that recommended in the guideline, no testing of the drug product for residual solvents needs to be considered. You need to a list of your actual solvents and respective limits. A list of the actual solvents and the respective limits are provided in the guidance document. Of note, there are 8 different documents pertaining to ICHQ3C on the FDA guidance page from 1997 to the 26th of May, 2020, and all are still in effect. All the additional ones are ones that added additional solvents to the list and their limits. So the next guidance that you need to look at is elemental impurities. There are 3 risk assessment steps. You identify known and potential sources of elemental impurities. You're going to evaluate for the presence of a particular elemental impurity in the drug product. You need to summarize and document the risk assessment. PDE is established based on factors such as likely oxidation state. That's your PDE is your permitted daily exposure. Likely oxidation state of the element in the drug product. the human exposure and safety data when it provides applicable information, most relevant animal studies, the route of administration, and relevant endpoints. And as we spoke about before, the FDA has issued a guidance document on the control of nitrosamine impurities, which was most recently updated in March of this year. And the update in March, it basically just extended the deadline to the end of March, which didn't really, at this point, it's a moot point. The FDA issued a guidance control of nitrosamine impurities in human drugs in September of 2020, and again, I'm sorry, updated in February 2021. This guidance recommends that manufacturers consider the potential causes of nitrosamine formation, as well as any other pathways observed, and evaluate the risk for nitrosamine contamination or formation in their APIs and drug products. The updated guidance discusses the unexpected findings of nitrosamine impurities in drugs such as angiotensin II receptor blockers, which are the ARBs I mentioned earlier, ranitidine, nizantidine, and metformin, resulting in the need for this risk assessment in any pharmaceutical product at risk for their presence. This updated guidance became effective the 31st of March, 2021. Risk assessments should be performed on both approved and marketed products and on pending applications for all APIs and drug products. Synchrogenic recommends our clients include a statement in the drug product and drug substance sections addressing the possibility of nitrosamine contamination. If nitrosamine impurities are possible, you need to include steps taken to mitigate this risk. So just a sentence that your process has been reviewed and determined that there are no nitrosamines or no way for nitrosamines to be introduced to the drug substance or drug product is sufficient for now for an FDA submission. The EMA has issued a similar guidance where the inclusion of statements regarding an evaluation for nitrosamine impurities and any steps taken to mitigate the nitrosamine impurities present is mandatory. As a note, if a guidance such as this one comes out and your product is already approved, you still need to conduct the appropriate testing under this guidance or the new guidance to ensure that it does not impact your product. The FDA has provided a link to their validated method for determining nitrosamines in the most recent version of this guidance document. The FDA's February 2021 guidance on nitrosamine provides a link. I just mentioned that. It's provided, the link to the validated laboratory method is provided as a footnote on page 2 of the February 2021 guidance. And this is the reaction that forms nitrosamines. And when you're discussing your impurity information, these are the locations where you're going to place it. For CMC modules, you're going to put it in 32S32, characterization in impurities, and that's for drug substance impurities. For drug product impurities, you're going to place them in 32P55, control of drug product, characterization of impurities. And this is for, again, impurities seen only in the drug product. And there are some out there. We have seen a few clients that have this situation. Your non-clinical modules where you're going to discuss this information. First off, your CMC modules are going to refer all your testing reports and everything go into the non-clinical modules. And then the non-clinical modules that are going to contain these discussions are module 2.4, non-clinical overview, and module 266, toxicology written summary. We have a list of the references that we use to write this portion of the presentation. And now we're going to go on to the case studies. During the case studies, the names of compounds discussed have been changed to protect the innocent. Any similarity to the names of our company or our parent company are purely intentional. Our next speaker is Dr. Angela Howard, who will be presenting our first case study. Dr. Howard is a board-certified toxicologist who has written non-clinical written and tabulated summaries among the many other documents that she's been writing. Dr. Howard?
Thank you, Nellie. Next slide, please. So this is the first case study. As Nellie pointed out, it's going to be of a fictional drug substance that we're calling Sotarant. And next slide, please. And so as we discussed, as we heard earlier, the definition of an impurity is any component of the new drug substance that is not the chemical as defined as the new drug substance. And that's from the ICH Q3A-R2 guidance. And there is a companion guidance, as Nellie pointed out, for the definition of an impurity in a drug product. But this case study will focus on the drug substance. And so for this, we have Sartarant. It is an oral drug. It will be given at 600 milligrams per day, and it is for an anti-tumor indication. Next slide, please. And so again, this is the decision tree that we referenced earlier, and we're going to step through this decision tree for this case study. Next slide, please. And so to begin, we start at the very top, and we ask the question, are the impurities that we have in this drug substance, are they greater than the identification threshold? And in this case, we know that we have two impurities. Impurity A is at 0.6% and impurity B is at 0.65%. And as Nellie indicated, there is an attachment 1 threshold where we will look to see if the actual level of our impurities is greater than the identification threshold. And we can see that that ID threshold is 0.1% or 1 milligram per day intake, whichever is lower. And so in this case, yes, both of our impurities are greater than the identification threshold. Next slide, please. And so as we continue down, we ask the question, do we know what the structures of those impurities are? And in this case, yes, both structures are known to the sponsor. And so we continue down the decision tree. Are there any known human relevant risks? And in this case, there were none. Next slide, please. And so as we continue down, then we have to ask the question, are the impurities greater than the qualification threshold? And so again, we have our levels that we know of impurity A, 0.6%, and impurity B, 0.65%. And if you think back to that table, the qualification threshold was 0.15% or 1 milligram per day intake, whichever is lower. And so yes, both of our impurities are greater than the qualification threshold. And so then we ask the question, can we reduce those impurities to not more than that qualification threshold? And in this case, the sponsor informed us that they cannot reduce the level of impurities to less than or equal to the qualification threshold. Next slide, please. And so since we cannot reduce the level of the impurities, we can attempt to qualify these substances based on the safety of the impurities, based on the general toxicity studies that were conducted. Now you'll see in this box below that it lists several types of studies. And while the guidance does not say that you must test the impurities in these studies, this would allow for the cleanest results. But in this case, the sponsor did not test the impurities. And therefore, we had to go through and calculate the amount of the impurities present in the drug substance that was used in the toxicity studies conducted. And so this is a two-step process. Next slide, please. And so the first step is to ask, are the impurities genotoxic? And we can answer this question in one of two ways based on the guidances, either through in vitro testing, either the Ames test or mouse lymphoma assay, or we can do some in silico assessments if the structures are known. In this case, we know the structures. So the FDA does require data from two complementary in silico methods. You have to use models that are based on expert rule-based models and statistically based models. And so the expert rule-based models use toxicological knowledge of structure and function relationships. The statistically based models will use statistical relationships between the effects of the compound and the physical chemical properties and structural characteristics. And so both methods identify structural alerts that are associated with specific toxicity with a specific molecular fragment. Next slide, please. And so in this case, the sponsor provided an in silico assessment per FDA guidelines, and they determined that both impurities were non-genotoxic. And so now if we comfortable that they're non-genotoxic, we can attempt to qualify the safety of the impurities by looking at the general toxicity studies in step 2. And next slide, thank you. And so we're looking to answer two questions here. The first is at what dose in the animal study are the impurities present at a higher level than the human dose? And so we're going to use the human equivalent dose in order to make this comparison. And then the second question that we'll look to answer is, were there serious adverse effects observed at that corresponding animal dose? So if you turn to the table, you can see that we have impurity A and impurity B, and the human dose we talked about earlier is 600 milligrams per day. And that gives us 3.6 milligrams per day of impurity A and 3.9 milligrams per day of impurity B. And so at the NOAEL, the no observed adverse effect level in the 13-week dog study, that value is 10 milligrams per kilogram per day. We end up with, once we do our conversion, 3.3 milligrams per day of impurity A and 2.7 milligrams per day of impurity B. And so in both of these cases, we have not given the animals enough of the impurity in order to be able to say that we don't feel there would be any effect in humans, given that we have a little bit of a higher dose. And so we actually went to the next dose in the study, which was the HNSTD, the highest non-severely toxic dose. And that was 15 milligrams per kilogram per day. And there we have 5 milligrams per day of impurity A that was administered and 4.1 milligrams per day of impurity B that was administered. And so next slide, please. And so to answer that first question, at what dose in the animal study are the impurities present? At a higher level than the human dose, we found that that dose was the HNSTD, so the highest non-severely toxic dose of 15 milligrams per kilogram per day. Oops, back one, thanks. And then, so once we established that dose, we had to ask, are there any serious adverse effects observed at that animal dose? And no, there were no lethal or irreversible toxicity effects noted for animals at the HNSTD of 15 milligrams per kilogram per day. And next slide, please. And so the key points to address in module 2 and 2.4, the non-clinical overview, and then the 266 toxicology written summary, based on the in silico assessment, neither impurity is expected to be genotoxic. We looked at the 13-week dog study, and we selected that for impurity qualification based on the level of the impurities present in the drug substance and the body weight of the animals, because remember, we're doing a human equivalent dose calculation. And the impurities are qualified based on the negative finding in the in silico mutagenicity assessment and by the level of the impurities present at the HNSTD in the 13-week repeat dose dog study. And next slide, please. And so I will now turn the presentation over to Dr. Rebecca Shirk, who will present the next 2 case studies. And Dr. Shirk is a principal regulatory writer with over 10 years of experience in non-clinical and clinical regulatory writing. Dr. Shirk.
Thank you for that introduction. Next slide, please. So for the last part of today's webinar, we're going to present 2 additional case studies based on our experience at Synchrogenics writing regulatory documents for new drug applications. Next slide, please. So the first case study we're going to present is for a drug that we're going to call Synchrogenic. And Synchrogenic had 2 manufacturing impurities that were observed in the drug substance, and we'll call them, not so creatively, impurity A and impurity B. So the first question we have to ask based on the decision tree from Q3A ICH guidance is whether these impurities are present at levels that exceed the identification threshold. The impurity A is present at 1.1% of the total peak area of the drug substance by chromatography, excuse me, and impurity B is present at 0.64% of the total peak area. So the identification threshold is based on the maximum daily dose, the intended maximum daily dose of drug substance. And in our case, Synchrogenic is going to be administered at 600 milligrams per day. So that takes us to the top row of the table. The identification threshold is 0.1% of the drug substance, or 1.0 milligrams of impurity per day intake, whichever is lower. And according to either one of these criteria, both impurities A and impurity B exceed the identification threshold. Next slide, please. So the next question is, are the structures known? And the sponsor successfully determined the structure of the impurities and accordingly did literature searches and in silico analysis to determine whether there was any potential for mutagenicity. And for both impurities, there was no known relevant risk identified. Next slide, please. So the next question is, yes, that's the correct one, thank you. Then the next question is whether the impurities exceed the qualification threshold. Again, this is based on the maximum daily dose. Both impurities do exceed the qualification threshold of 0.15% in the drug substance, or 1.0 milligrams per day intake of the impurity. So according to the decision tree, obviously the most expedient route is to try to reduce the impurity levels to less than the qualification threshold. And in this case, yes, the client successfully was able to reduce the level of the impurities in subsequent batches of drug substance used for clinical studies and for the registration batch. So the the impurities are not believed to pose a risk to humans. Qualification of safety for human use is not needed. Next slide, please. However, a question remained. Did the impurities present in the toxicology grade batches of drugs that were used for the non-clinical testing, did they impact the safety profile of the drug? So a qualification study was indeed conducted. They did a study to compare the toxicology profile of the drug substance with impurities versus without impurities. So this was a 2-week repeat dose study in rats. One treatment group received a toxicology grade batch once daily for 14 days, and the second treatment group received once daily treatment for 14 days with a clinical grade batch with greatly reduced levels of impurity. Next slide, please. Next slide. So the outcome of the study was that the toxicology profile was comparable between the two treatment groups in the 14-day rat study. So these results were discussed in detail in Module 2.4, non-clinical overview, and Module 2.6.6, the toxicology written summary. And they stated, the sponsor was able to state that the 14 daily doses of synchrogenib at 25 mg per kg with manufacturing impurities, in other words, the toxicology grade, or without manufacturing impurities, the clinical grade, produced comparable toxicity profile between the two treatment groups. So the sponsor could conclude that no adverse findings in the toxicity studies were attributed to an impurity. Next slide, please. Our last case study is for a drug that we'll call Allcurcet. Next slide, please. So Allcurcet had 4 observed and potential impurities that we'll call impurities A through D. The structures were known, and so database searches and literature searches were conducted. In addition, in silico predictions for bacterial mutagenic potential were evaluated using 2 methods, the expert rule-based and the statistical-based methods, to look for structural alerts, that is, molecular structures that are associated with mutagenic activity. According to the ICH M7 guidance, the impurities can be classified with respect to their mutagenic and carcinogenic potential as class 1 through 5. Class 5 has the least amount of risk. They have no structural alerts, or a structural alert is identified, but there's additional laboratory data to demonstrate a lack of mutagenic activity. And at the other end of the spectrum are class 1 impurities that are known to be mutagenic and known to have carcinogenic properties in vivo. So we're going to now step through the process the sponsor followed for these 4 impurities. If we go to the next slide, please. So they're summarized in the next few slides in a tabulated format. If we'll start on the left-hand side and work our way across the table towards the right-hand side, we'll see what steps the sponsor took and their ultimate classification and the decision. So impurity A went through in silico predictions for mutagenicity, and in both types of analyses, impurity A came back as negative. Therefore, no further Ames testing or any other types of mutagenicity testing were required. And impurity A could be classified as a class 5 impurity without, in other words, it has no structural alert, and therefore it could be treated as a non-mutagenic impurity. So the sponsor decided to control this impurity to not more than 0.1% levels in the drug substance, which is safely below the levels of the qualification threshold. Sorry. In impurity B, it came back positive from in silico predictions of mutagenicity. However, the structural alert was the same as the alert that was present in the parent drug, Allcurcet. Allcurcet, of course, had been previously tested, was known to be Ames negative, and therefore impurity B required no further testing. It could be classified as a class 4 impurity, has an alerting structure, but similar to the same one as the one in Allcurcet, which is known to be Ames negative. Therefore, it could be treated as a non-mutagenic impurity, and the sponsor determined to control it to levels not more than 0.1% in the drug substance. In other words, below the qualification threshold. Next slide, please. Okay, impurity C. Impurity C also came back with in silico predictions of mutagenic potential. If they had stopped evaluating it at this point, it would have been classified as a class 3. Mutagenic, that had mutagenic potential, unrelated to Allcurcet, without any mutagenic data. However, of course, they moved forward doing Ames testing. And in this case, impurity C came back as Ames positive. So this impurity needed to be classified as a class 2 impurity. It's a known mutagen, but without any known carcinogenic potential at this point. So the sponsor needed to take the route, according to the ICH guidance, of controlling it to levels below the acceptable intake of 1.5 micrograms per day for a drug that will be administered for more than 10 years. And I should have mentioned at the outset, this is a drug that is for chronic treatment for pretty much the lifetime of the patient. So they needed to control the impurity levels below an acceptable intake of 1.5 micrograms per ml based on the threshold of toxicological concern method. And because Allcurcet will be dosed at 70 mg per kg, they were able to, they determined that they would control it to less than or equal to 20 parts per million, which is the same as 0.2 micrograms per mg times a dose of 70 mg per day, brings them into a maximum maximum intake of 1.4 micrograms per day of impurity C over their lifetime. So drugs that, impurities that are classified as a class 1, class 2, or class 3 must be limited to an acceptable intake. And that's the approach that they took for impurity C. Next slide, please. The last impurity that we'll discuss is impurity D. It also came through that it was positive by both methods of in silico prediction. The structural alert was unrelated to the structure of Allcurcet, so at that point it was considered a class 3 impurity. However, in vitro Ames testing was conducted and it came back as Ames negative, and therefore the impurity was reclassified as a class 5, meaning it had a structural alert, but there was additional data demonstrating a lack of mutagenic or carcinogenic potential. So being treated as a non-mutagenic impurity, sponsor determined to control it to no more than 0.1% levels in the drug substance. In other words, less than the qualification threshold. Next slide, please. So these outcomes for all 4 impurities were addressed in the module 2.4 non-clinical overview and the module 266 toxicology written summary. And the authors needed to carefully describe all of the impurities and state that the class 2 genotoxic impurity that was identified as Ames positive will be controlled in the drug substance to a level that's appropriate for a potential genotoxic impurity based on the maximum daily dose. The class 4 and class 5 impurities will be treated as non-mutagenic impurities and controlled in the drug substance to a level of less than 0.1%. And the final thing that they needed to clarify in their submission documents is that carcinogenicity studies had not yet been conducted, at least at the time of the NDA submission. But they had committed to completing a 2-year carcinogenicity study in rats and a 6-month study in mice as a post-approval commitment, and as had been previously agreed upon with the FDA. Next slide, please. So that's the last of our case studies that we wish to present. Again, the key references, the ICH guidances that are followed in evaluating these impurities, at least in the case studies, were ICH Q3A for drug substance, Q3B for drug products, and ICH M7 for mutagenic impurities. And that brings us to the conclusion of our presentation. We can now open the floor for questions.
Alrighty, okay. So we have our first question here on the list. Asks, if the structure of the impurity cannot be identified at NDA submission, would control below qualification limit be acceptable at the NDA submission? For example, if M7 cannot be followed.
So this is Angela. I can take that question. I think that it would have to be controlled below the identification threshold at 0.1%. Looking at the decision tree, if it cannot be controlled below that, then you go immediately to that box of studies that the FDA would like you to conduct in terms of making sure that the impurity is safe. And remember, you can always also, I think, call a meeting with the FDA to kind of discuss what issues you think might be there and get their view on what the best way to solve that would be as well.
Wonderful. Okay. This next question, I wasn't sure if this is one you would like to answer, but I'll ask it anyways here. This says, thanks for the good presentations. One question on the API specification. If we use a reagent, for example, dimethyl sulfate, in the last step of synthesis, do we need to include it in the API specification?
I'll take that one. Unless you have a discussion with the FDA, otherwise, I would include that on the specification. Again, you can discuss it with what controls you have, but since it's the last step of the synthesis, you really can't, there's no purge points after that. So it may be something that you would want to control.
Alrighty. Looking at the open questions we have here, if a potential M7 impurity with a limit of 60 parts per million is consistently controlled at levels below LOQ less than 5 parts per million, should a specification be set?
If it's controlled, what data do you have that how is it controlled? Is it controlled as an in-process control? If that is the case, then, and you can demonstrate that is where it's controlled, then that would be fine. But I know the FDA is going to want to see some sort of control to say that you are controlling it. You know, it's not just, your specification is a demonstration that your process, everything has run smoothly for each and every lot. And if you see something unusual appear, then on the specification, then that's an indication that something was amiss when the manufacture or the synthesis of your drug substance.
Nice.
So you're going to want to have something on there, either a in-process control or your drug substance control.
Okay, and it looks like there was a follow-on comment saying no IPC testing of 10 historical batches.
This no testing or no positive results. Sorry.
Let's see if there's a response there, but okay. We'll move. In the meantime, we'll move. Testing is less than 5 parts per million. Okay,
so it is probably in process for 10 lots. Then I would present that as your way of controlling it. You're not controlling it on the finished product. You're controlling it in process.
Okay. Next question we have here are leachables and extractables considered impurities?
Yes.
Wonderful. Okay. Next question we have here asks, in your example where you ran the tox studies using 2 different lots, plus or minus impurities in question, did you find that this approach was preferable to FDA than isolating the impurities and then testing those directly in a tox study? Certainly easier for a sponsor. Also, any advice on how long the study should be to qualify impurities?
So I can try to answer that. In this case, well, in general, the FDA does not require that the impurity must be synthesized and tested in a pure form. So in this case, the type of qualification study they did really was to address a non-clinical question as opposed to address potential safety concerns for a drug substance that would be administered to humans. I do know that this submission went through and the drug was approved without any questions about their qualification strategy. So in this case, it did not appear to cause any concerns.
All righty, wonderful. Our next question we have here asks, once drug product impurities have been identified, do the impurities controls in the release specifications, and stability specification needs to be identical? Thank you.
If there is if you can demonstrate your rationale for an increase for stability, then no, but you still have to have it controlled below the ICH guidance thresholds, even for stability.
Okay. The next question we have here, how to address degradation impurity that is the product of the reaction between excipients?
You should have sufficient data by the time of NDA submission to have an understanding of how much what this impurity is going to be in a normal situation at the end of shelf life, and the FDA is going to want you to be really close to that. They're not going to, you're not going to have a whole lot of wiggle room. So you want to put it at, look at it realistically at your data. Give yourself some leeway, based on some scientific rationale of why you're allowing that much leeway, and make sure that your level has no human toxicities.
Wonderful. Okay. Let's see. And if you'd like, just a reminder, if you'd like to have some questions answered, just click the Q&A button there at the bottom to submit those. We have just a few more minutes. We'll go through some more of these. Let's see. We have the next question asks, how many synthesis steps away from API do you consider the impurity would be low enough so that it would not be included?
You need to do fate and purge studies to determine that. If it's the step before and you're, or the 2 steps before and the step before, you can demonstrate that it's adequately purged at that point, then you're fine. If you can't demonstrate that it's purged, then you need to have a specification for it.
Okay. Next one asks, for nitrosamine risk assessments, if we concluded that a confirmatory testing is needed, should we submit to the NDA or inform FDA, or wait until confirmatory testing is completed?
That depends on your time. If you're able to complete your confirmatory testing before the NDA submission date, then I would go that way. You also, I would encourage that to be one of the questions that you ask the FDA during your pre-NDA meeting, and show them what data, include in the briefing book what data you have to support your position.
Okay, let's see here. The next question we have is a genotoxicity assessment done for all observed impurities, regardless of being above or below the thresholds?
You want to do a structural comparison. Look at all the structures that are there that for the impurities that you have, and if any of them rise an alert, that's when you want to continue with the next step for the genotoxicity determinations.
See, we did have a question asking about access to the slides. The link to access that is within the chat. So we'll just go ahead and address that way. Okay, we have time for, I believe, one more question here before we conclude. Just asking, are there conditions under which the threshold of toxicological concern approach should not be used?
Rebecca or Angela, do you have a?
I believe, oh, go ahead.
Yeah, I was just going to say, yeah, I think for the TTC, I mean, it's used even for FDA, even within those that are considered genotoxic, as long as you're below that 1.5 micrograms per day. Rebecca, is that your understanding as well?
I believe so. I think the issue is if you actually come back with positive data in an in vivo carcinogenicity study, then you're going to have different requirements for what's an acceptable level.
Yes, that's right.
Righty. Okay, so we are at time. So to ensure that everyone's schedule is aligned, in case you have another meeting coming up, I will begin to conclude today's